Hello, in this video we will discuss about the tyrosine kinase receptor. So the receptor tyrosine kinase are the high affinity cell surface receptor for many polypeptide growth factors and cytokines as well as hormones, which that is give the signal to the cell. So the receptor tyrosine is basically the dimer, and basically it will remain detached before the signal, before the binding with the ligand. So after the binding with the ligand, it will become a dimerization. So we will discuss about that things. First is the monomerization to become a dimerization. So here is the receptor tyrosine kinase is the phosphorylated dimer activated by the binding of the ligand, and the tyrosine is the basically the amino acid which that is bind with the phosphate group. So the 6 phosphate group will be bind to become a dimer activated through ATP use. So let's begin to understand about the ligand. Ligand can be a cytokines and hormones, growth factors, or other polypeptide, or on the other hand, the other a pathogen can be, and which can be detected and engulfed due to the it's not harmful if the pathogen. On the other hand, if not harmful bacteria, basically not pathogen. If pathogen, that means it is different. So here is the ligand, which basically exocytosis and proliferate and growth will be occur. So after the exocytosis of the some material from the cell can be happen due to the tyrosine kinase activity. So here is the tyrosine kinase is the extracellular domain and intracellular domain. Extracellular domain contain a receptor for the ligand binding receptor, and after the here is the gamma monomers which that bind to become a intracellular domains which that is bind with a phosphate group to become a activate, and in this way the RAS protein is present. Basically the RAS protein is the used for the GDP inactive to become a GTP, and the RTK is used for the activation of the tyrosine kinase receptor, and the RAS become a GTP. So the phosphorylase, and the here is the growth factor receptor bound protein 2 GERB2, and source is the basically the saver of soul bind with it, and in this way here is the soluble RAF. So RAF will be soluble RAF will become a membrane bound RAF due to the activation through a RAS. After the activation, GTP is used for it. And in this way, here is the mitogen activated protein kinase. And in this way, it will use for it to RAF to activate the mito MEK protein inactivate to become a activate, and this MEK phosphorylase through a mitogen activated protein kinase to become a mitogen activated protein kinase, and again will convert into the mitogen activated protein kinase is to become a ARC activation, and this ARC is the transcriptional factor bind within it in the nucleus. After the binding with the MAP mitogen activated protein kinase to use to ARC, which protein will bind with the transcriptional factor to proliferate and translation to become a proliferate cell, differentiate cell, and cell growth. So this is the basic general structure. If we will discuss about the insulin, so the insulin is an anabolic hormone which that is released due to the glucose level high if the blood hyperglycemia will stimulate the insulin, and insulin will bind with the insulin receptor. So the insulin receptor is the tyrosine kinase also. So here is a dimer. This dimer will become combined together with the phosphorylation, the same mechanism, but in this way the insulin substrate 1 are present. In this way here is the red color is showing in the figure to understand. So in this way the insulin receptor substrate receptor will phosphorylase through and bind with the phosphoinositide 3 kinase protein. And after this binding with phosphate group to become other phosphorylation of the kinase enzyme, and on the other hand the other pathway, the mTOR pathway and other pathway can be occur, and this pathway will transcription signal. On the other hand, here is the PDK1 pathway will be activated through PI3K, and PKB pathway will be activated, and other signaling pathway will be activated. And in this way, here is the cytoplasmic vesicle with glucose transporter will be activate and bind with the plasma membrane is here. You can understand as well. On the other hand, the transcription factor from the other signal pathway for use for the GLUT4 channel production, growth, as well as the lipid phospholipid of bilayer phospholipid for the GLUT channel vesicle formation, and this GLUT channel used for the influx of the glucose, inflow of the glucose or influx of the glucose. So this was the insulin sensitivity, which that is the tyrosine kinase receptor are present, and same mechanism in the pathogen interaction with the dendritic cell. After the pathogen interaction, recognition and endocytose the dendritic cell to produce the interferon gamma. This interferon gamma is basically the cytokine, and in this way this is the cell mediator, which that is the immune cell mediator, and in this way it is used for the activation of macrophage by the binding of the tyrosine kinase receptor and natural killer cell and neutrophil. So on the other hand, it will immature to become mature. How it's possible? Let's begin to understand. Here is a dimer which that bind with the interferon gamma. After this binding, there are many cytokines, but here I am taking the interferon gamma. So the Janus activated kinase protein will bind both tyrosine domain, and in this way here is the intracellular and the extracellular used for the binding receptor protein. So the STAT will be bind after the phosphorylation of the JAK, and after the JAK will be phosphorylation, and the STAT also phosphorylation through ATP, and in this way the JAK will be remain backside, and in this way here you can see the dimerization will be occur, and this dimerization basically the STAT come together to bind to each other through phosphorylation, but the JAK will be phosphorylation and dephosphorylase to become capture another interferon gamma, while here is the dimerization will be occur. After the dimerization of the STAT, this STAT will be dimerized and go as a transcription factor with bind with the DNA of the nucleus of the cell, for example the natural killer cell and macrophage, and after this it will be growth, proliferation and maturation. After the transcription to here the polymerase enzyme and helicase enzyme used for it to produce messenger RNA through transcription to become a translation in the cytoplasm through ribosome, and this growth factor as well as the proliferation, maturation and other mechanism will be occur. So this was the tyrosine kinase receptor. So thanks for watching. Please make sure to subscribe, like and share.